Good afternoon, everybody. Thanks for coming to today's Dean Lectures. And for those of you who've been here before, you know that this is a venue for us to celebrate the achievements of faculty who are either appointed as professor or promoted to professor. And, you know, as I always say, and I'll say it again, is that it's not easy to get on the faculty here. You have to have outstanding promise to be invited onto the faculty. But it's even more special to be promoted to the rank of professor. And it's a decision that's not made by me or really by the A&P committee. It's made by peers around the world who we seek their opinion and they write to us. And although I see our faculty every day and get to talk to them, I learn a lot from reading those letters. And it's always impressive to me to see the high esteem that our faculty are held in by their peers around the world, and that's definitely true for Danny. And I always say too that it's kind of it's a little silly for me to be introducing our professors to you, since many of you have worked with the speakers for many years. But I'll just say a little bit about Daniele Fallin. So Danny came to our school in 2000 after completing her PhD in genetic epi at Case Western University in Cleveland. And did you work with Elston? Who was your? Yeah, but before that, she had been she was at Florida, undergraduate at University of Florida, and then had spent some time at the University of South Florida. And here she heads the Center for Autism and Developmental Disabilities Epidemiology. And she's done a lot in her career. She's looked at the genetics of diseases associated with aging and chronic disease, but also has had an incredible impact in autism and works with a group of epidemiologists, statisticians, our colleagues down the street at KKI, people who can phenotype autistic spectrum disorders, and to look at the genetics of developmental disabilities broadly. And you know, her CV, as you probably know, is a mix of really high impact work that's at sort of the tail end of finding genes for diseases, but also she works with colleagues in biostatistics and elsewhere to do methodological work too. So an incredible broad array of work within her field. And like many genetic epidemiologists, she's really sought after as a collaborator because she has such a incredible insight into the methodology and works so well with people. She directs our Center for Autism and Developmental Disability Epidemiology and also heads the genetic epi concentration since 2007 or so. She was promoted to associate professor in 2005 and then recently to professor. So I think Danny is somebody that I rely on before she was professor, and now that she's a professor even more, I get to rely on her for her expertise and her advice about things related to developmental disabilities in children. And unfortunately, it's a growth area, as she knows better than I. You all saw the CDC report from just last month. It says that 1 in 50 or so boys in the U.S. has an autistic spectrum disorder. It's just hard to believe. And so the kind of work that she does, where she looks not only at the genetic epidemiology, but also has the epidemiological skills to look at environmental and other factors, just couldn't be more important. So Danny, congratulations on being promoted. We're looking forward to hearing your talk about epi. Okay, so.
Thank you for that very nice introduction, and thank you all for taking the time to be here. So I'm going to start by just laying out what I'm going to talk about today, which is, as Mike said, I'm trained as a genetic epidemiologist, so one could think very deterministically that is all of us walking around with G's, T's, C's, and A's determining everything about us. And hopefully, we all know that that's probably not the whole story. And as a genetic epidemiologist, I would have forever argued that probably there are multiple causes, including multiple genes, maybe environments, maybe the interaction between genes and environments, and maybe through pathways that might not be direct, like through susceptibility to infection or some environment that ultimately leads to a disease. And I would still stand by this kind of thinking, but what I'm going to talk to you about today is that about 6-ish years ago, I kind of expanded this thinking to add the idea of epigenetics on top of genetic epidemiology. And my thinking there was because all of these mechanisms that I, say that I'm interested in, how genes relate to disease, how environments relate to disease, and how the two of them together may relate to disease, really depend on regulation of those genes and expression of those genes in a way that is controlled by many things, including epigenetic mechanisms. And so with that in mind, my sort of thinking has expanded a bit to not only the genetic epi-training that I had, but trying to inform myself about epigenetics, put together then the epidemiology, the genetic epidemiology, and the epigenetics world to move forward. And so that's the perspective that I'm going to hope to bring to you today. And so I won't get to talk about several of the other things that I do in autism specifically, but I did want to start, before I get into the epigenetics, by saying a big thank you to those who work with me in autism. And so this was a picture from World Autism Day, which was earlier this month, right here in this building. And I think having this crowd, which is only a subset of the folks that were there that day that were part of this picture to show the support for the School of Public Health more generally in supporting Autism Awareness Month. But specifically, all of these folks work on studies with me, trying to understand both the genetic and the more broad epidemiologic perspectives and risk factors and causes of autism. And so I'm not going to highlight everybody individually, but this does consist of several coordinators that really have made my life just unbelievably wonderful lately. So Jamie Dahm here, Carmen Berry here, and Michelle Landrum have really led the rest of this team. Chris, I'll mention in a minute in another way. And so even though I don't get to talk as much as I could today about some of the particular studies that they're involved with, I want you to know that there's this really active group of really dedicated staff here working on these things. And Mike right in the middle helping to support all of this. I also want to make sure that before I get going on the epigenetics sort of message I want to talk about today, that I thank everybody in my quote-unquote Fallin group. This is really my students and postdocs. And so you'll see, a crowd here going from my postdocs down to my students and even some of my master students that are just about to graduate, both of which did autism projects that I'm very proud of. And the most important group, the Herfel group, which is my husband, who's in the audience, but for some reason didn't want me to put a picture of him in the bathtub on the slide, and my two sons, Bryce and Kellen. And then also a lot of this thinking, actually all of this thinking really has been spurred from an interaction I had about 6 or 7 years ago with Andy Feinberg, who is here in the School of Medicine, actually housed over in the Rangos building, who leads, we put together, he and I and Rafa Arzari and Carl Broman, who was actually here at the time, an application for a Center for Excellence in Genome Sciences called a CEGS. And the one here is dedicated to really pushing forward epigenetic mechanisms and ideas in common disease, and this integration that I'm going to talk about today between what we think of as phenotypes or disease, genotypes or kind of sequence level variation, and epigenotypes or epigenetics, which are these other things that I'm going to define a little bit more for you. And there is an entire crowd of folks that work through the SEGS and with Andy's lab, and also with Rafa's lab here in the Department of Biostatistics. And so this really is a cross collaboration between folks in the School of Medicine and folks here in multiple departments in the School of Public Health. So themes in epidemiology that I think are really relevant to themes in epigenetics and in genetic epidemiology. You can see how I wanted to come up with some clever title because they all sound the same. So the first is environmental causes. So I'm going to spend just a slide or two telling you why I think this is a natural place to put together the environmental cause kind of field of epidemiology with the molecular and the genetics group. And that's because there's growing evidence of environmental susceptibility to epigenetic marks, and therefore these might be an underlying mechanism or mediator of how environments manifest risk for disease. And then I'll say something about age-related changes, which is also provocative in the sense that a lot of diseases, now autism may not fit into this as much as some of my work in the other end of life in aging diseases, but the idea that although we have the same sequence from birth, we definitely don't have the same manifestation of disability until later and later in life. And in fact, lots of what we all study in this room has higher and higher risk as you get older and older. And so understanding how age plays a role in that biology would be important, and I think, again, epigenetic mechanisms might help us in that regard. And then lastly, genetic causes. So this is kind of the bread and butter of what I do and what I know best, and so I've come up with multiple ways in which I think genes might be part of the story, and I will hopefully dissect those for you a little bit better. So when Andy and I first got together to sort of think about these things, he had a student, Hans Bjornsson, who's now practicing physician across the street, and at the time he was an MD PhD student with Andy, and he was very interested in pushing with us this idea of how you would incorporate kind of the common disease, common variant hypothesis that was happening in genetic epidemiology at the time with this idea of how epigenetics plays a role. And so we kind of sat together and said, okay, well, let's just write out some sort of framework. What are we even talking about? What kinds of relationships do we even mean? And maybe you can dissect them a little bit and then try to answer them piecemeal before kind of tackling this thing in a very fuzzy way. So we came up with this framework that I've changed multiple times since, but I've been heartened by the fact that we now are starting to see that pieces of this were probably a good bet, at least a good starting point. And so I'm going to walk you through this framework as it was in that TIGS paper, just because I think it's helpful for you to get some insight into how I've been thinking about these things. And so the top part right here is what I was already doing. The idea that there are, genotypes or genetics that can affect risk for disease, and that there are environments that can affect risk for disease, and that there's some interplay potentially between these two. So that's kind of what I'll think of as traditional genetics or traditional genetic epidemiology thinking. And I'm certainly not here to say that any of that was wrong, but rather to expand that thinking beyond that. And so really we're going to talk about the rest of that graphic. But to do that, everything that comes below that point is going to be things that happen through epigenotype or as a consequence of some interplay between genes and epigenotype. I better define a little bit more about epigenetics. And so here's where I was going to show my teaching skills too, because in 754, which is a epidemiology course that I co-teach with Tom Glass and Brian Lau, we have this year integrated something called Poll Everywhere to make our teaching more interactive. And so we actually have these great slides now where in real time we put up a question and you guys either text or put on your computer through the website the answer. And then we can see the growing yeses versus nos and whether there's really debate in the room or whether everybody says, oh yes, that's obvious, Dr. Fallin, move on. And it's really great. So right here I was going to have one of those polls that said, how many people know what epigenetics means, so that I could sort of judge my audience of who does and doesn't. I think that would be fabulous to start doing in all my scientific talks, right? So you know where really to pitch. But alas, I didn't get it together to make it so that the transition would work easily. Next time. So what is epigenetics? You'll see lots of definitions. I'm not trying, for those of you who do practice this field, this may seem a little bit baby talk to you, but I'm hoping that it's enough so that all of us will be on the same page. But the idea is that this is genetic information that is not contained in the ACTG sequence. So the typical, what you've learned about probably in your high school classes or your New York Times articles or wherever now, even if you don't do this kind of work, you've seen that genes are made up of these letters that spell sort of coding for what makes up the protein or the working parts of our bodies. And that's typically what we think of, the sequence, as being genetics. So I'm not talking about that sequence, but rather other kinds of chemical modifications to that sequence that can still be inherited through cell replication and still carry information, specifically information that will regulate how and when those genes are turned on and used by the body. And so let me give you a little cartoon that I borrowed and I think is referenced on the next slide. This is that sequence that I was telling you about. And physically, this sequence is actually wrapped up in really, really, really tiny bits and mushed together. And so if you unpack that a little bit, I just want to show you that as you unpack it, the first thing you can see is that it's not shown on here, but sometimes when there are C's, there's a methyl group on top of it. And so on the next slide, I'm going to show you that that's one chemical modification when that C has a methyl on top of it that we call DNA methylation. There's going to be another type of modifications that these things that the DNA is wrapped around ultimately are histones and then make larger structures. They too can have chemical modifications. Let's just say acetylation for right now. And all I want to show you in this slide is that if you look at this one at the top, it's kind of really spread out, right? So if I were some kind of mechanism that was going to come in and read sequence and sort of read and say what should be done next, I'd want that to be spread out so that I could access that little black area where the sequence is. Versus the one below it right here, where everything's scrunched up. And if I were trying to come in and read, I wouldn't be able to read that because everything's really tightly together. And so the sort of heuristic to that is when things are exposed like this, those things can function, you can read that and then ultimately translate it into a protein. When it's tight like this, it's hard to get in, you can't read it. So what does that really mean? Well, this is that same slide, but now with a little bit more context around it. Heuristically, that means in this case, you're going to have higher expression because the architecture that needs to be there to read that and ultimately make a transcript and then make a protein can get there. Down here, That's not the case. And so what do those correspond to in terms of epigenetic marks? Well, remember I told you could have Cs that were methylated or not. For those of you who like the chemical version of that, that's this part right here. Here's a C, methylated. Here, it's white versus red. So when red is on there, that's hard to access, right? So that's this idea of lower expression. When white is on there, that's easier to access. And then I also talked to you about these chemical modifications of those histones. That's this little part right here. Look right here. Well, think about that as sort of pushing out, right? So it's keeping things a little bit separate from each other. And again, things can come in. So that's the idea of high expression versus when those are gone, things can get really close together. So when those are gone and things are close together, that's kind of like low expression. That's all I'm going to say about that, but it's just to give you a sense of why this is a relevant thing to think about, because then it has consequences in terms of whether or not you have a particular sequence that has any consequence will depend on whether it's actually expressed and made into a protein or not, right? So this heuristic, I think, is the important thing for my talk. And I've talked about this in two ways now, that epigenetics is this sort of inherited information not contained in the sequence but has something to do with regulation, and I've mentioned DNA methylation of cytosines. I've also mentioned histone modifications like acetylation. Epigenetics is really expanding. More and more is understood about these mechanisms, and so that's not the only two situations that might come under an umbrella of what's called epigenetics. And so this is a slide that my postdoc Chris Costa put together, and it really shows the methylation that I was talking about, things that can methylate and demethylate. Larger structure that I didn't mention may also come under this umbrella. So really, although I've talked a lot about histone modifications and DNA methylation, I'm really picking pieces of a larger story that come under this umbrella of epigenetics. So back to my framework. So the idea I mentioned before is what was up here was already what I had been doing and others have been doing, and then what's below is trying to integrate some other thoughts with this. And so the first thing is just proof of principle. Is it really possible that changes in these epigenotypes, if you will, so these chemical modifications that are not the sequence itself, can they manifest risk for disease? And there was already lots of literature to say yes. In fact, they can. So I'm not going to show you good examples of that, but there are several genetic disorders that we know are consequences of differences in methylation, or imprinting disorders, which also have the same epigenetic context. And so I think we can trust that first little part of this framework. And you can think, again, just sort of superficially why that would be. Well, here's a gene that has a lot of methylation, and so whatever architecture needs to get in there to express that gene and ultimately create a protein really can't get there. So it's sort of a low expression state, right? Here's one where the opposite is true. Things can get there, and this is what we might think of as a high expression state. Well, for the same DNA sequence, if this was something that wasn't supposed to be expressed very often, and all of a sudden you start expressing it willy-nilly, that could be a problem, and that could manifest a biology that then becomes a risk for disease, and vice versa. So it makes sort of face sense that that would be the case. So themes in epidemiology. Now that I've sort of explained what I mean by epigenetics, hopefully in not too long, and hopefully, but at least with enough that you can understand where I'm coming from to go forward, I want to tell you a little bit then about these themes and each one of them and why there's some growing evidence that makes it provocative to think about integration. So I'll start with things that I think we want to think about. If we're interested in the fact that there might be some epigenetic relationship to disease risk, right, then we better understand what things control variability in epigenotype, if you will. If there's variation between people that's somehow related to their variability and risk for disease, then what explains that variation? That's part of our story, right? That's the ultimate cause that we're interested in. And so there are at least this list, probably more now, but I'll just highlight to you the ones that we highlighted at the time. And that is to think about environment in the person that we're talking about right now, age, which I'll mention again, parental epigenetic state. There's still a lot of discussion about whether or not these things can go cross-generationally. Environment of the parent. This is also part of this provocative thinking that there's certainly evidence in the parent, but even in grandparents and others, whether those environments may manifest changes that ultimately lead to risk. And then whether genotypes themselves control variability in epigenetics. And I'm going to go through these in more detail. I'm just sort of laying the landscape for you. So let's start with the environmental ones. And so one proof of principle thinking, and there are growing stories like this, but one just to give you the flavor that this too is not outside the realm of possibility, is the story of agouti mice. And so through multiple, this is just a review, but through multiple primary papers, these mice, you can see some are sort of yellow and very large, some are darker color and sort of normal weight, and then some mottled things in between. And these mice, so we would say that those are different phenotypically, both in terms of obesity and in terms of coat color. And those mice have the same DNA sequence. The difference is something that has changed this phenotype that is not the sequence itself. And so by experiment, it has been shown that environment can manifest that kind of difference. And environment in several different ways. The mom's diet, in terms of methyl donors, pharmaceuticals, particular things given to the mom like valproic acid, and even maternal behavior, whether you're licked and loved or whether you're isolated as a pup. All of those things can manifest this kind of phenotypic change. So those are environments that are leading to this change. And it turns out the difference is it's leading to change through difference in epigenetic marks and mechanisms in these animals. So that's one proof of principle that this idea of going from environment through epigenotype can affect a trait difference or a phenotype difference. That's in animals. In humans, about 5 years ago now, there was a provocative study that looked cross-sectionally at twins. And so these aren't the same twins followed for 50 years, but rather looked at 3-year-old twin pairs who were of the same genotype, so identical twins, and say 50-year-old twins who were of the same genotype, so identical. So they didn't have any variability in sequence of DNA, but you could look at their methylation patterns or their histone acetylation patterns or epigenetic patterns, if you will, and see whether those were the same or not. And what they saw was the 3-year-olds looked pretty similar. The 50-year-olds didn't look so similar. So something was happening with age, either accumulation of environment or aging itself or something about both, but that was sort of provocative that environment may matter, or in fact, in some sense, epigenetics is changing over time because they were so similar at young ages, and this is just a metric of that similarity, and not so similar at older ages. So that was cross-sectional. That was not in the same people. I'll mention again when I talk about age what you might want to do if you want to look in the same people sort of longitudinally or prospectively over time. So environmentally, we've seen in the animals that diet and some of these other things like nature and nurture might matter, or sorry, nurture might matter. What about in humans? So there have been studies looking at diet in humans, and so I'm not going to spend too much time on this slide, but just remind you that, and for some of you who may never have had to learn these things, this is a one-carbon transfer pathway, and the amount of folic acid and other methyl donors you take in will, if you follow this through, affect the methylation that's available for DNA methylation. And so you can imagine how diet supplying this will affect sort of the sustainability of DNA methylation. And so a lot of thought has gone into how dietary factors may be one of those environments that then goes through epigenotype and then ultimately to a phenotype. But all sorts of other environmental factors have already started to be looked at in humans: endocrine disruptors, metals, infections, and I'd point you to a recent issue in the International Journal of Epidemiology that was dedicated to integration of epigenetics and epidemiology. And so there are several papers about this issue. It just came out last month that I'll mention. But one of them was this: for the environmental epidemiologists in the room, you might want to take a look. They basically talked about the environmental chemical exposures and the ideas of how epigenetics might be useful if that's your interest as an epidemiologist. And there's sort of key messages where there's certainly growing evidence that these things might be susceptible to epigenetic change, or epigenetic mechanisms might be susceptible to environmental toxicants, and that you should really think about how to integrate that. And then things even like nurture that we saw in the animals have started to be looked at in humans. And that same IJE issue was one of a couple of different recent articles on how SES position seems to be correlated, at least. And this one was early life SES and in its relationship to adult level DNA methylation. And that has been really, I think, interesting and provocative for the psychiatry and behavioral epidemiology crowd, thinking about ways in which you can get these sort of social and behavioral experiences that then do manifest in real disease outcomes. How is that happening? What is the biological mechanism? And I think there's a lot of hope that maybe this will shed some light on that. So that's environmental causes integration. What about age-related causes or changes? And in my mind, this is just sort of fundamental to understanding, are these things labile over time? Do they really change with age? And that could mean that it's about the aging process, or it could mean that it's cumulative exposures. And there's growing evidence that these things do change with age. So I mentioned to you a few things already, like that twin pair study. But there have also been multiple reports in the last sort of 5 years of cross-sectional studies that show correlations between things like DNA methylation and age. And then we set out in a study that I'll say a little bit more about soon, an Icelandic cohort, to look at changes in the same individuals over time. So rather than having to rely on these cross-sectional kinds of reports, if we had DNA samples over multiple time points in the same individuals, can we see for that person that they're actually changing over time? And we did that by looking at something that we call global methylation, which is just what's happening overall in your genome without knowing where in the genome, just a sort of bucket approach, versus site-specific. If you can look at particular genes, do you see changes either increasing or decreasing in methylation, in this case, at those sites? So that's what this global versus site-specific means. And we did, in fact, see both. So when we did the global approach, this is a little bit funny of a figure, but I just want to show you the red first. What's on the left is methylation change. So if there was no change, that would be kind of the 0 per person. And then you can see that if people did change, increased in their methylation over time, it would go this way. If they decreased, it would go this way. And again, this is that bucket approach, their overall genomic methylation. And what you can see is in red, if you sort by change, yeah, there's a several group that decreased a lot. There's a lot that didn't do much, and then there's a group that increased a lot. And the key question there would be, well, how much of that is just variability in the assay or something like that over time? This is about 10 years apart. And that's what all these other lines in between are. That this is what you would expect just by that kind of variability. And you can see that the red line does separate from those pretty well. So that does argue that there does seem to be real change going on, but in fact it's not in one direction or the other. That sort of bucket approach is seeing big changes in some people, and that's kind of overall. So that's useful as a proof of principle, but it's not satisfying in that you don't know where, or you don't really know how to interpret that beyond face value. So more recently, we looked at the same individuals, but used an approach that could actually look across the genome, but at gene-specific sites. So see, at this particular spot in the genome was a person getting more or less methylated over about a 10-year period. And in that case, what we saw, so this is, this was going from 0, increased methylation, decreased methylation. Now I've shifted the axis to that. So now 0 is kind of right in here. And so some of this variable, remember everybody kind of changed a little bit. So this isn't exactly 0, but this is still the change metric. And most people, or I shouldn't say most people, most sites now had an average change of sort of nothing. So most sites in the genome were stable. They weren't actually changing over this period. But if you look at this black line right here, boop, back down. It does look like there's 2, maybe even a third here, bump. And these folks are ones that do seem, this is the absolute value change, so we don't know which direction from this figure, but there does seem to be this second hump of folks that, or I shouldn't say folks, second hump of sites in the genome that are changing. So what we learned from this is you can't just say, yes, DNA methylation changes with age, or no, it doesn't. It's like every other epidemiology answer, it depends. It depends on where you are in the genome. And I think that can be useful as we think about how to use this downstream, but it was an important foundational point to make heading in, that it's not an all or nothing kind of story. There do appear to be places in the genome that are stable, at least over this 10-year period in these people, which are older people. And then there are other places that do tend to be changing quite a bit. And that might be useful if you're trying to look for things that might manifest a change. Why look at spots that don't tend to change over time? Maybe you want to reduce yourself to looking at those places in the genome that are changing over time and might therefore be susceptible to these things. So age-related changes. It does look like there are changes. It is not stable everywhere, but the places that are changing over time might be particular. What about genetic causes? And so I already mentioned the genes that have some manifest risk on disease, and I'll introduce this subscript idea. G sub IND just means genes that are independent of what I'm going to talk about next in terms of how they affect risk. That's, I think, different than genes that might manifest through an epigenetic mechanism. And so that's why I call these G sub EPG, for genes that really control epigenetic variation itself. And so these are really controlling variability between people in their epigenotype, which then ultimately has an effect on disease. And there's some proof that that's true. These are 2 different recent studies that have shown, if you look on the left, going from bottom to top is methylation amount, and going from left to right is genotype at a SNP, which is a spot, for those of you who don't do genetics, a spot in the genome where there may be folks who have 2 copies of a sequence that has a C on it. Some of you may have one from one parent that was a C and from another parent that was a T, and others of you may have both parents gave you a T. So I can classify you into one of three categories at that spot in the genome in terms of your sequence by these three groups. And when you look at those three groups, this group has sort of a low methylation status, this middle group has a middle methylation status, and this group has a high methylation status. And this group saw that for several places. A different group recently reported seeing a similar thing in their samples. Now it's not quite as dose response, but you can certainly see that one genotype has a very different methylation pattern than the other two. And this is not unique in the genome. There are many places where that occurs. And this is too much to go through in detail. I want you to get the big picture context. Bottom to top is genotype position. It's these Cs, CTs, and TTs idea going through the whole genome. Left to right is methylation going through the whole genome. And the circles and the size of the circles is correlation or association between those two things. So something like this would be a highly associated thing because the methylation is correlated highly to the genotype. Okay? And so you can see that when you're in the same part of the genome, these things sort of light up. It does look like nearby sequence variation is controlling epigenetic variation between individuals. But in fact, also off the diagonal looks to be true. So these things are true, and they tend to be, a relative proportion, it looks like, of what's going on with variation in epigenetics. So I've mentioned how environment can manifest it, how age can manifest it, and now a good example from real data that genes can manifest this kind of variability. So I think our framework is playing out to some extent in thinking about all of those simultaneously. Let me say something about then the third way I think genes might play a role. So G independent, gene that controls epigenotype and therefore has a risk, and then G that I've called DEP for dependent on epigenetic mechanism. And this is the one actually that got me really interested in doing this because it's the one that I worry we've been missing all along. And because this is really the idea where I think genes and epigenetics come into play as an interaction, or even environments and genes come into play. Let me unpack that a little bit for you. So what I'm really talking about is this idea that there's a risk, an epigenetic risk that might manifest disease. There's also a genetic risk that might manifest disease, but that genotype is only a problem under certain levels of control from the epigenotype. So here's an example. This is a C normally in a person. If that was mutated to a T, or for whatever reason was a T in some subset of people, and that T is a problem that creates a problematic protein in some way, well, right now, with that high methylation state, this is a low expressed area. And so that's not going to have much consequence on disease risk because it's hardly ever expressed. But if for whatever reason there was inter-individual variability, so some of those people with Ts had low methylation, or you had exposure that changed the status, or age changed the status, any of those reasons that I just talked about, now you have a situation where this gene is expressed, and that T is expressed, and if that T is problematic, now it's relevant. Now it matters. And so in that context, knowing both the sequence variability, so this genotype-dependent idea here, and the epigenetic state would be important to understanding that risk. So one other way to think about it is here. So this is a sequence that's now not the same letters, but now it's an AG. So this person here has an AG genotype. So does this person. But this person, because of the methylation state, is actually sort of expressing themselves as a G. This person, same genotype, but because of the methylation state, is expressing themselves as an A. And so if G was problematic, these folks may end up at higher risk for disease than these folks. Whereas I, as the lowly genetic epidemiologist doing this study where I've measured the AG genotype, wouldn't have ever seen this difference. So that's a concern. Here's how it actually did manifest in some of the work that I've done in several psychiatric disorders. We have cases represented here. And so these are two boys, one affected, one not affected, and his dad and his mom, and this one's dad and mom. And these are just, for symbolism, their genotypes. So they all have an AB. And this child, though, has an AB and is unaffected. This child has an AB and is affected. And why would that be? If I were doing a case control study, I would have said, counting up all the kids that are cases, check for a heterozygote, counting up all the people that are controls, check another heterozygote. And if those were sort of balancing each other out, again, I wouldn't see this risk. But if I saw that, in fact, this child got that A from his dad, but this child got a B from his dad and got the A from the mom, and the one from the mom turned out to not be the one that was expressed, so this person is looking like an A and this person is looking like a B genetically, then I could have separated out these kinds of phenomenon and seen something. So we call parent of origin analysis, where I paid attention to which parent gave a particular allele. And that's how you could help to see these things. And so we and others have done that. I'll start on the right. We did that in autism through a linkage analysis, and you can see a couple of places in the genome. I'll just orient you really quickly. This is position on the genome going from left to right. This is some sort of statistical support going from bottom to top for whether or not there's a genetic location for autism in that part of the genome. And then what I'll show you is in blue is if that was inherited from the dad. In red is the evidence if it was inherited from the mom. And we found several places in the genome where it looked like it was parent of origin specific in that inheritance. You can see that's when there's separation between the blue and the red lines. Don Arking, who's here in Institute for Genetic Medicine, was part of one of the first genome-wide association studies for autism. And in their study, they reported associations with something, with a gene in the contactin family. And he also saw a parent of origin effect in his first initial discovery of a SNP association in this gene. I'm just going to orient you to the blue, which is the boys who were affected with autism. And what they did was say, okay, those who have a TT have an increased risk. That's why this is greater than this dashed line. That's their estimate of risk. But if they got a T from their dad, their risk looked about like baseline. In this case, if they had gotten the T from the mom, it looked like it was an increased risk like these homozygotes. So that fits with the story I said on the previous slide, that had you lumped these two together, those effects might have washed out and you wouldn't have seen much. But if you said which parent contributed that allele, you can separate those who tend to be at risk versus those who don't. So this sort of theme has been true certainly in psychiatric genetics, but potentially more broadly. In fact, I've done some work in other non-psychiatric disorders and seen a little bit of the same thing. So in terms of genetics, at least 3 different ways that I think genes can play a role here. Independently, through an epigenetic mechanism because it actually controls epigenetype, and then also dependently because it's part of some interaction. And in fact, I think this is where the typical maybe epigenetics is helping us with gene by environment interaction might be the most salient. Because if it were an environment that is manifesting risk through this pathway and changing sort of the regulation of some genes, if those genes themselves have genetic predisposition to be important or not depending on that context, that's the route for which that could happen. So that might be one important way to understand these things. Okay, and lastly, I don't mention this very often because it's not part of what I do, but this certainly all so far has been talking in the world of sort of etiology and cause. This area of epigenetics, especially in cancer, has already been pushed well forward in terms of just thinking of this as a biomarker. So if epigenetic marks are a consequence of disease rather than part of the cause of the disease, they may still be very useful as biomarkers of things like prediction, diagnosis, prognosis. And this is a little bit old of a review, but I think it just gives you that idea that there's certainly ways to measure epigenetic patterns that can help you to understand thinking about early diagnosis, prognosis, and maybe even follow-up or particular treatment options. So that biomarker world is also an application of this work and of this field that's very relevant to epidemiologists. So promises and challenges. Well, I think hopefully I've convinced you there are causal mediation modifier roles and potentially also biomarker roles, and for all those reasons we should understand what we're doing. But it doesn't come without major questions to still be answered. Some are foundational, just where are the spots in the genome that have some variability in methylation that we should even target and look at? How frequent are they? Are people really only different when they are different by like a couple of percents on a methylation scale, or 20 or 30 or 40%? You know, what are we looking at in terms of this kind of distribution? And then what about the contribution of genes versus environments to that epigenetic variation? Is that important? And if so, how would we apply that in our studies? And that leads me really to thinking about disease-related issues. I think it's important that we go in a priori and make some, sort of begin thinking about whether we're asking this as a potential causal role, as a mediator, as a modifier, or simply as a marker. Because once we get these sort of correlative answers in a lot of the designs that are being used, we won't be able to answer that post hoc. So going in with sort of a framework in mind, I think, is really important. Tissue relevance I'll bring up again in terms of foundational, but that's another one that sort of crosses both the just general and the disease-related domains of questions that we need to answer. And then major challenges, tissue availability. So that sticks to the relevance. I'll say a couple more things about that in a minute, and then I'll mention some of these other measurement design and statistical problems. So the disease-related questions, well, why do I think that's important to do as you go in? Because I think once you see these kinds of results, you're going to then have to try to explain them. And if you don't have some sort of theoretical framework going in for which of these explanations is most useful to you, or some way to dissect one versus the other, you might get stuck. So I think it's really important to think about that going in. For example, remember I said we found that there are spots in the genome that looked like they were stable and not changing over time, but other spots that were changing over time. Remember this idea? So if I were to filter the genome and only look at those spots that I thought were changing over time, that may help me to decide which ones of these I'm going after, or vice versa, deciding which one of these to go after. Maybe I'm interested in those that are mediators of genes. Well, your sequence level DNA doesn't really change over time, so maybe these that are stable over time might be the better subset to think further about. So you can imagine there's just different ways you could go about the design based on your underlying hypothesis. So the major challenges, though, are not few. This is a really nascent field, so there's a lot still going on to be figured out. And one that's hotly debated is tissue availability and relevance. And so, as Mike said, and as I indicated at the very beginning, I'm, probably most interested in autism and the causes and consequences of autism. And in that case, we think that the primary tissue of affection is brain. We do not have a lot of brain samples in life. We certainly do have some post-life. But there are a lot of issues to work out for any kind of question about characterizing autism and risk factors when you think about autopsy samples versus in-life samples. But if we're forced to in-life samples for the kinds of studies that we're doing that are asking questions about environment and genes and other things going on simultaneously, we'd want something that you could do in an epidemiologic study. And in that case, we're stuck with blood for the most part. So the real question immediately begs itself, is blood a decent enough proxy to ask these kinds of questions? And that is, I can tell you right now, there is no right or wrong answer. And this is hotly debated at meetings, on conference calls, all in study sections, all over the place. And there are good arguments on both sides, and I think data will tell us, because a lot of us, including my group, are moving forward with the blood to see what we can see. And we feel armed with that based on this list and a growing one. Hopefully we're going to write a little commentary soon about this with some folks across, actually, the world working in autism epigenetics. And the idea is that we are seeing that measures of expression and even measures of epigenetic marks do distinguish some disease, non-disease, and there are correlations between periphery methylation and primary tissue methylation. It will not be one-to-one, for sure. That's by nature of the whole role of epigenetics in determining where and when things are expressed. But it looks like there are hallmark features, so at least a subset of things that might be important will be seen in blood. And so we and other groups have taken the sort of mantra of, let's look at both. Let's look at in-life samples that have all the positive things we want to think about in terms of understanding etiology and the kinds of epidemiologic designs we're doing. Let's also pay attention to autopsy samples and see what kind of concordance we get between the two and try to move forward informed by both perspectives. But it's an open area of debate. Catalog of information, as I've mentioned, is a little bit tough because we need to know where in the genome to be looking for these things and what we should expect to see in terms of variability in those regions. Measurement, also a big issue. I've been a little loosey-goosey about that. I've mostly talked about DNA methylation, which is here in green, partly because that's one of the things that's most easily measured in the sort of high throughput way compared to the gestalt of different kinds of epigenetic marks I mentioned. So it's not that other things couldn't be measured, but really the epigenetic applications in epidemiology have been with DNA methylation so far. And at that, it can be global. That was that bucket approach I mentioned. It can also be genome-wide and site-specific, meaning it's genome-wide, but you actually know where in the genome you're looking. And in that case, you could do whole genome bisulfite sequencing. That is still fairly expensive and time-consuming. Most of us are doing genome scale, but gene-specific methylation assays, and these are primarily array-based. We've been doing CHARM that was developed by Andy Feinberg group, but also the Illumina 450K platform. And then you could certainly follow up things, or if you had really good hunches to begin with, start with particular genes and do another sort of bisulfite sequencing, but at a smaller scale. So you can see still work needs to be done in that arena. Design, I've mentioned a little bit. It's different than the world of genetic epidemiology that I was raised in, because your genes are your genes are your genes, whether you got my DNA today or you get it when I'm 55 or you got it when I was 25, for the most part. Epigenetics, as I've said, it looks like there are some things that are changing with time, not necessarily the case. So timing will matter in a way that maybe we genetic epidemiologists had gotten a little lazy about in terms of biosampling. So we're more apt to learn from our colleagues in environmental epidemiology about the key issues when time matters. And then in terms of statistical modeling and all those things, we're more apt to learn about how to do that from folks where that kind of change over time is more relevant. And that leads me to the statistical approaches and integrations, which is what I'll end on with some examples. I just want to say that there's been a lot of talk in the last several years in epidemiology journals about this integration. I'm certainly not the only one thinking about these things. I thought this was a great picture from one of those reviews a couple years ago, sort of talking about the points that I just mentioned, both the biology of why this could be relevant to questions of etiology in terms of different places in the lifespan, which places in terms of cells and tissues might be relevant in some sense, but more about timing. And then what kinds of risk factors one might be thinking about influencing those at those different windows of time. And then the cold water of what we can actually measure. And so a lot of what might be very interesting is in here, and we may be resorting to animal models for that kind of work. And then here, the pragmatic epidemiologist has to think about what you can actually do and what you can learn based on what you can actually do. So last but not least, statistical approaches and statistical integration. And we are blessed by having Rafa and Jeff and a ton of other people that I could list, a very long list of folks in biostatistics, in oncology, and in the SEG specifically, who have been thinking hard about these approaches. And so what have we done? Well, we've tried to approach multiple designs with the goal of having prospective designs for some of the question reasons that I mentioned about whether you think it's causal or mediated or a biomarker, et cetera. Measurement, we've really tried to stick to genome scale, gene specific, so mostly CHARM, although some 450K. And then statistical approaches, there really has to be a beginning point. What are you going to try to filter the genome on to start with? What are you looking for? And so I'm going to tell you just a little bit about two things, thinking about things as variably methylated regions versus differentially methylated regions. These are two terms that are now being used more and more in the field. And our focus really is for either of these to ultimately answer questions about correlations between methylation and phenotypes or methylation and exposures. So what is a variably methylated region? Well, here's one way to depict methylation in a region. So this is a position going left to right on the chromosome. And then this is methylation going from 0 to 1 on the bottom to top. And then each one of these lines is a plot from one person's data. And you can see there's not a lot of variability here, right? They all look pretty tight, as opposed to this, where now there's a lot of variability in this section between people, right? That's variation across people, or a variably methylated region, because it's a whole region that looks like that. And so one way you might think about is these are the places, this is where the money is, right? If there's no change between people, if things look like this, then there's not really an exposure or a trait or anything that's going to correlate well with that, right? But when they look like this heterogeneity could be explained by underlying differences in these people that have to do with exposures or ultimately have to do with diseases that they are at risk for. So one way to think about this is how do you filter the genome to pick out these things statistically? And so Jeff Leake's group and Andrew, who was a student of mine while he was a PhD student, came up with a method to do just that. And so you can see that there's a way to sort of capture that variability and plot it and then designate a region to specify as a variably methylated region. That's opposed to a differentially methylated region, because remember here, I haven't talked about any particular disease or any particular exposed, not exposed groups. This is just variation, right? As opposed to differential methylation, which is specific to some factor. So in this case, same kind of picture. Left to right is position on the chromosome. Bottom to top is amount of methylation. Each dot is a person. So these are lots of different probes along this chromosome. But what you can see is that in this region, those in red, which are dots from cancer samples, look very different than those in blue, which are dots from non-cancer samples. And so we would say this is a DMR. It's a region of differential methylation. In this case, it's a cancer DMR because the differential part comes from being different by cancer. Okay? VMR, DMR. That's the difference. So I'm just going to hopefully very quickly go through an example from two different studies that I have helped run. And so one is this Iceland study that I already mentioned in terms of the age work. This is part of the Ages Reykjavik study, which is a very long cohort since the 60s in Reykjavik, Iceland, that is now an aging cohort because only those who have survived that long are still left. And we had DNA samples from a visit that happened in around 1991 and a second visit that happened between 2002 and 2005. And these folks were fairly old. Visit at age 6, so this first visit was already 75. So these are elderly folks that had a mean of 75 and then moved to a mean of 85 at the next visit. And they were about 11 years apart. And our questions really fit that model that I showed you before. It's kind of been modified a little bit, but the idea is, are there epigenetic changes, so an epigenome may be a modified epigenome at the second visit, that then manifest some quantitative phenotype or disease? And are those things themselves affected by DNA variation, and are they affected by exposure, environmental exposures? And when thinking about it this way, if we go back to that idea of VMRs and DMRs, these right here are those measures of the epigenome. We have to think about how to filter those to best inform us about whether there's a change over time, and then ultimately whether they're related to phenotypes or whether they're related to exposures. And so if you think about it that way, what we did for this particular study was we started by focusing on variably methylated regions. So those spots where we think there's some meat because there's some variability between people to begin with. Then amongst those where there's some variability, we asked about change over time. Are they really changing from visit 6 to visit 7, or are they same? So cross individuals and within individuals. And then amongst those VMRs, we looked for DMRs, places that were differential because of some trait. And in this case, we focused on BMI, as I'll show you. And now currently we're doing the same strategy, but for exposures. So just to give you a little picture, if you think about methylation in the whole genome, we want to think about ways to focus on the sites that we think are relevant for our question. So VMRs is one way to filter that. Of those variably methylated regions, some of that variation may be explained by a difference due to cancer, or in this case, due to BMI or to an exposure. Those would be those differentially methylated regions. And all of that is variability across individuals. Okay? But then this time thing also comes in. We want to know, at a spot that we know now is variable across people, is it also variable within that person? Is it changing over time as they go through life? And so there's a subset that are also variable within people. So we're trying to keep straight those different kinds of questions. And remember, this was the idea that there are spots, so this section that was variably methylated, that's these right here, right? Some of those were changing with time, some were not. Changing with time, not changing with time. Were the labels dynamic, stable? So in that, we did identify VMRs. We did see that there were some that had changes over time and some did not. And then we focused on those VMRs in this case that did not change over time. We're thinking, is this something that's like a signature about the person? It's not changing because of exposure or other things. It's more like a characteristic of the person. Do we see any of those where there's variable methylation that's related to a health-related trait like BMI? So remember, we looked at the VMRs. Then we wanted to find those DMRs, and so we thought about, of these VMRs, let's only look at those that are stable and not worry for this question about those that are changing over time. And what we saw is this. Methylation bottom to top, position going left to right. We saw those in blue were those that were characterized by BMI as obese. Those in red, each individual dashed line as a person, were those characterized as normal. Those solid lines are the sort of the averages for those two, and you can see a separation in this region for normal versus obese. This was visit 7. This was the 10-year earlier visit 6. And if you think about taking that region then and just getting a sort of average methylation for each person across this region, you could do a scatter plot like that, and you could see very strong correlation at these spots between BMI and methylation. So that's one way to go from whole genome, variably methylated regions, to differentially methylated regions. One more example. DMRs. We could take the approach of going straight for differentially methylated regions, not take that first filter approach, but go straight at the whole genome, just looking for those DMRs. And that's been our approach in another study that I'm involved in that is directly related to autism ultimately. And that's one where we really focused on studies that have information about young children. Two of these studies, the early study, which I'm a co-PI of, and the National Children's Study, which is a national sample, both have followed women through pregnancy and through that baby's early life, and actually even beyond 12 months. But for the sake of these epigenetic studies, we're very interested in what's happening to methylation of the mom, which is what we can measure during pregnancy, and what's happening to the mom and the child in terms of methylation after pregnancy, and how those might correlate to both environments and to phenotypes. So that's this sort of light bulb idea, is putting together the epigenotype, the environment, the phenotype, and in the context of genotypes. Okay. And early, which I'm very proud of, is a 4-site study that I've been involved with. It's not like NCS in that it's an enriched cohort. These are moms who already have a child who has an autism disorder, who has decided to become pregnant again. So these children are at a higher risk than the general population for becoming autistic. And so we can follow them, sort of the natural progression of what happens to them in early life, ages 0 to 3. So that gives us lots of developmental milestone data, but it also gives us pregnancy data in terms of asking questions about environment and epigenetics very early on, prospectively, rather than having to ask about it once they're diagnosed. And so we thought about lots of things we could ask in terms of relationships between environment and epigenome, between epigenome and phenotype, and how genotype plays a role in that. And I'm just going to tell you about one that's based on cross-sectional data we have from a third study, which is called 3, which is just on newborns. And so we're going to start by looking not at VMRs first, but take the whole genome and try to go straight to these DMRs. And I know I'm running a little long, so I'm not going to spend too much time on this, but I want to give credit where credit's due. This is not a trivial thing to think about doing genome-wide association. It's very similar in a sense to genome-wide association of SNPs, but now we're talking about genome-wide association of methylation. So looking for these differentially methylated regions, if it were case control, is kind of like doing a case control GWAS. There are millions to look through to come up with a list that you can have some statistical statement about. And so Rafa Irizarry and his group with us, and again Andrew, who was my PhD student at the time, had a paper, I guess we all had a paper in the same IJE last month, where they really had an elegant way of going about this. And all I want to show you is to remind you, this is that DMR that I showed you before. Any one of these spots, any one of these positions is a probe. You could just set up that probe and say, okay, what do I expect the DNA methylation value to be at that probe as a function of the trait of interest. In this case, I'll talk to you about gestational age in a moment, but as a function of gestational age, and adjusting for something that will determine batch effects and try to get rid of some of the measurement issues that happen in array data. And what they did was say, okay, let's just fit that model for every sort of column on these kinds of data, and take these betas, which will be something about that difference, and plot them. And then once you have each one of those spots has a difference that you can measure, then smooth. And once you smooth, you could come up with a region by saying, okay, I am going to say everything outside the 99th percentile of this is something that I'm willing to call a region. So this right here would be what we would call differentially methylated, because all those beta values tend to be much lower or more extreme than the general difference between two groups. And then this amount in green right here would be the area, and that's one way to measure the statistical metric of this region in terms of its magnitude. You're not done. You've checked off all the way through identifying places that have this kind of differentially methylated region assessment. How do you know what in the heck to do with that in terms of, is that going to happen by chance? Is it not going to happen by chance? What kind of statistical support do I want to put on that uncertainty about that question? And so they developed also a permutation procedure that will handle that. So they developed a way to permute things so that as if there were no association between this region and your trait. Well, I'm going to keep using gestational age because that's the result I'm about to show you. If there were no difference between this region and gestational age in terms of methylation, I could shuffle the data to create that kind of situation, and then I could see whether I got results similar to the one I see in my real data or not. And if they look very different than what I would have expected by chance if there was nothing there, then that's better support to me, statistical evidence anyway, that there really is an association there. So that's the method that we applied in looking at, remember I said for this project we're interested ultimately in longitudinal information, but as kind of a first step, we looked at pilot data that was just cross-sectionally, methylation and a birth outcome in the baby. And what we saw was indeed you could identify these places where there did seem to be differences in methylation across babies as a function of their gestational age at birth. And this was based on 141 that were through the 3 study here at Hopkins that was led by Lynn Goldman and Frank Witter and Rolf Holden when he was here, and several others. And so we were able to identify these areas. So this is that magnitude of this sort of green area. And then this is where it is in the genome. And then these are the p-values and q-values, and these are already genome-wide. So this is not like in those of you who do GWAS where you then have to say, why isn't it 10 to the negative 8? This is already accommodating that multiplicity. This is what it looks like if you look at the data. I think it's pretty cool. So if you go from left to right, its position, bottom to top is methylation amount. And what Andrew did was then code these, Andrew and I should say Wajin Lee, who was on this particular analysis, they looked at binning gestational ages at birth by color. And so you can see that through this whole region, there's a really strong sort of dose relationship, with the youngest ages at birth being these sort of higher methylation and the oldest ages at birth being down here. Very consistent across that region. If you were to average across that region and look at the correlation between methylation and gestational age, this is the kind of relationship you would see. And then Andrew asked the question, well, what do adults look like? You know, are we just picking up something about what's happening as you get closer and closer to full gestation? Do we have a sample of adults we can look in to see if A, are they variable at all? Because clearly these guys are variable, these babies. But are adults variable? And if so, or if not, where do they lie on this distribution? And with the hypothesis that maybe they're around here with what happens when you get to the later gestation periods and sort of the movement is done. And in fact, that's right. We looked at about 150 adults. And you see just that. They are not variable. They tend to all be about the same, and they tend to be at these extreme or the 41 weeks, sort of the end of that period. And if you look at all three of those that I showed you on that table, they all have these very nice dose-response relationships, and the adults always end up just past the blue, just past the 41 weeks. That is not true in the rest of the genome, by the way. That's only true in these sort of top hits of ours. If you go down longer in the list, you lose this thing that it looks like adults are at the end of it. And these have some implication for interest for further work in terms of the consequences of gestational age in early childhood and even in adulthood. Maybe this will help us to understand the biology a little bit better. I think that's work to still be done, but it's certainly proof of principle in our larger planning for this grant that we can do these kinds of questions and sort of pick apart pieces of that overall set of goals. So, in the end, I think there are really roles for a cause, for mediation, for modifier, for gene by environment or environment by epigene, et cetera, interaction. I think you can't be too loosey-goosey, though. You have to talk about which one of those paths you're asking a question about at the time. Try to spell it out so that then you can communicate it better at the end. We are not without major questions to still be answered, both foundational and with respect to disease. And we're not without major challenges. This field is not settled law at all. There are still lots of things that are to be determined and that could be shaped and refined in ways that we haven't even thought of yet. But I hope that I've argued to you that integration of these ideas into our perspective of epidemiology could be really useful and could lead to new insights. So what do I think is needed? I think we need more studies that do all three of these. I think we need to clearly specify hypotheses. We need to work with our partners on measurement tools. We need to work with our biostatistical partners on analytical tools. We need to think about longitudinal data when we can. We need to think about different age windows. A lot of that work that I showed you was either at one end of the age spectrum or the other end of the age spectrum. There's a whole lot in the middle, it turns out. So we need to think about how to apply that. And partnerships with basic scientists. This is not going to be integration with population science and done. We really need to understand, especially because there's a window of opportunity we can never really get at in large epidemiologic studies. We have to work with our basic science partners to back and forth understand how to refine our questions, what we can answer, what they can answer. And so I would encourage that to happen even more than it already is. And I acknowledge everybody up front, but I didn't say much about early NCS and 3, so I want to make sure that that doesn't go without saying that there were a lot of people that did a lot of work to get those national children's studies and those 3 samples in place. And so I really appreciate them and certainly all the folks that have participated in all these studies that I have the great opportunity to be a part of. So thanks to all of you.
Danny, stay up here. You have to, we're filming this. We always film these, but we're especially filming this for your mom. So we want to make sure you stay behind the podium and that everybody uses the microphone so that we can get it on tape. So I'll start with the first question. You showed that analysis. You described it very briefly where you talked about, that they could come up with a distribution, Rafi, based on what you would expect with no association. So is there the equivalent of disequilibrium between in methylation? So if this cytosine is methylated, is the next one more likely to be methylated?
Yeah, so equivalent is probably not the right word, but analogous, yes. So it does appear that things that are close together are correlated with each other. And in fact, that smoothing idea partly borrows from that. And methods for lots of, and there are folks in the room, even more expertise. I look at Casper and others out in the audience that could say even more about this. But it does look like across array methods and maybe even sequencing methods that borrowing information like that is really important.
Right. So if do you use a Bayesian approach or?
You can use things as simple as just a straight up smoother, and you can use things that are more sophisticated. And I think that's part of the biostatistical techniques that are being developed that are somewhat platform based and situation based, but not always.
It seems like you would get more power if you used Bayesian. So questions or comments? So let me ask a second one then, because you said sort of in an offhand way that it's being argued about whether the epigenetics are inherited.
Yeah.
But we know it is. I mean, if you look at tie-ins and ligers and hennies versus mules, I mean they're clearly inherited in a consistent pattern in those examples, right?
So it depends on the definition of inherited. So certainly in life, as cells replicate, these patterns are inherited. The question becomes about cross-generational inheritance. Right. So one argument would be, you saw me present things that said that genotypes are part of the story of variation in epigenotypes, right? So in that sense, if you did a heritability analysis on it, you should see that they're heritable because they're controlled by genes, at least to some extent, right? I think where in my sort of basic science colleagues, if they're in the room, can step in, but I think where the real challenge gets tricky is trying to explain a biological mechanism for that transgenerational passing of epigenetic marks has been elusive. Anybody want to add to that? Okay.
I have a different question, Danny. There are some genes that are associated with disease. To think of BRCA1, BRCA2 as an example, or ankylosing spondylitis and B27, HLA-B27. I was wondering if those are, I guess they are independent genes, right? Is there any variation among those genes in terms of VMR, DMR, or overexpression, underexpression? Yeah, among those who have the gene, no disease, have the gene, have the disease.
Right. So I think, homie, what you're asking is, are the same genes that we know to be part of the biology of a disease because we know about sequence variation or mutations that can cause that disease, are those same genes the ones that are implicated if you look at methylation variation? Is that what you're asking? Yeah. And so I think the short answer is we don't really know. We, as one of our filters, often look for that. When we see, what rises to the top in our epigenetics analysis, we say, well, have these genes been implicated in previous GWAS or other kinds of sequence level data analyses, or have they been implicated in other kinds of biological analyses? And that would sort of lend further evidence to the biology. But whether, the same genes sometimes have mutational differences and other times have methylation differences that manifest the same phenotype, I'd say there probably are several examples of that, but I don't know that that's a standard assumption yet. Yeah.
Oh, thank you. So a question about autism and biomarkers that may be expressed as a result of these changes in the transcriptome, presumably. Do you see, in the autism studies, are you seeing gene products that are circulating that are different in autism that can be linked to these differences in genetic expression?
Yeah. So it would be wonderful to have a very clear biomarker of autism. Right now, that's certainly not the case. If you asked me, can you use expression profiles in the periphery to discriminate cases and controls? Maybe a bit. Can you then define which pieces of that profile pattern are the actual genes that are causing that discrimination? No.
Or do you see a metabolome that's different? Do you see?
Yeah, not yet, but I think lots of people are thinking that way. And for me, the challenge there is, so is that helping me phenotypically just to have better classification, or is that telling me something about the etiology? Right? Because those could just be biomarkers that are consequences of what's happening in these kids. And so I still have to grapple with how would I use that information. But yes, certainly that's being done right now by us and many others. Yeah. There's a couple more back there. Yeah. Okay.
I see one hand. Is there another hand?
There was one in the center and then one in the back.
Maybe you could go get that mic up there.
Hi, thank you very much for the talk. I had, I guess, a brief question regarding the lag or latency between methylation patterns and disease manifestation, because it would seem like to me, whereas a short latency period would be good for, utility as a marker of disease, in terms of identifying risk early, you'd want to see a big lag time. Yeah,
so I think that timing question is critical. And that's part of the, like I said, the cold water of that figure from the AJE review. If timing, where it is really critical, is well before things that we can measure, we may have missed it. I think the gestational age work that we did shows that had we looked at those same things, maybe we don't have these data yet, but had we looked at them in 1-year-olds or 10-year-olds, would we still have seen those as places in the genome that might be relevant to some subsequent biology? And I don't know. And so I think that's a critical thing. We may be missing things because we aren't getting the right window of opportunity. Yeah.
My question is, you had talked about mid-onset diseases, and I was wondering if anyone was looking into autoimmune diseases for epigenetics, and also since autoimmune diseases tend to have a skewer in terms of gender towards women, if there's really any difference in any epigenetic differences in the sexes.
Yeah, so great questions. So to answer the first one, absolutely. I'm involved in several, and I know of many that I'm not really involved with at all, that are looking at all kinds of different disorders, including autoimmune disorders, for these kinds of marks. And in some sense, that's a little, I wouldn't say more exciting, because I think what I do is exciting too, but that is potentially the target tissue. So blood might be even more relevant than in the kinds of things that I do, where I'm using it as a proxy, because it's probably not the primary tissue involved. So it can be very interesting for those reasons. The question about sex is a really good one. And although I still don't think, well, nobody's really written a paper specifically on sex, but if you start looking through the literature, you'll see that there are these sex differences reported. And we have certainly also seen that that is an important factor. And so it's possible. In the autism world, it's the opposite. The ratio is 4 or 5 to 1 for boys versus girls with autism. And so we have a little working group that Autism Speaks supports, where we talk monthly about, and I really mean everybody. There's people in Canada and in the UK and here and all over the place. We just brainstorm about what could this be, what could we do from an epigenetics perspective. And so there's really a lot of enthusiasm that there may be some thinking in epigenetic mechanisms that might help to explain that boy to girl ratio. But that's still, I think there's a lot of ideas that are not necessarily complementary yet, a lot of different things that aren't consistent across that we're still trying to work out in our discussions. Yeah.
Thanks.
Okay, I think it's about 1:15 or so maybe we'll carry the discussion out to where the wine and cheese and stuff is. And Danny, I want to thank you for a great talk.
Thanks.